Good morning. You guys behave so well for Dr. Roberts. It's my pleasure to be here at this excellent meeting, and I have the pleasure of introducing Dr. Saumya Das, who's both an MD and a PhD. He's Associate Professor of Medicine at Harvard and Director of the Resync and Advanced Therapy Program at Mass Gen. Runs a nice basic science lab himself there, looking at things like biomarkers, which really interests me. But he's made the translational step to be co-founder and principal scientific and medical advisor for Thryv Therepeutics. Welcome.
Thank you very much for that kind introduction, and thanks to the organizers for having us. Thryv Therepeutics, which spun out of work I'd done about 15 years ago, is also proud to kind of help sponsor this meeting, so really happy to be here and share some of the work. These are my disclosures, and of course there is a conflict because I founded Thryv and I consult for them, but so what you'll hear, take that with a grain of salt, I guess. So I wanted to talk a little bit about the science behind what we are trying to do. This is, I think, a unique approach to treat arrhythmias. And it started with work looking at a kinase called SGK1, serum and glucocorticoid responsive kinase. And this is a somewhat unique kinase because at least in the heart, it is activated in conditions of stress. And I know people don't like to see Western blots and things, but you can see the one below, that's from human patients. And you can see that at baseline, the levels are quite low of the phosphorylated form, that's the active form of the kinase. But in stress conditions like heart failure or hypertrophy, it gets markedly upregulated. And so this sort of led to a series of experiments that was published in 2012 that led to design of mice where it could activate this kinase and study what it does, or inhibit the kinase and see whether there was some effect, some beneficial effect of inhibition. And you can see that we designed these mice quite well. You could get an increase in activity of these kinase in the hearts of these mice specifically. And when you looked at these mice that had activation of the kinase, on the right-hand side, you can see ECGs from these mice. On the top is a wild-type control mice, and on the bottom is a mouse with activation of this kinase. These mice were really prone to arrhythmias. And mice generally don't have ventricular arrhythmias, so that was somewhat unusual. These mice, when you stress them, either by putting catheters in the heart and stimulating them or subjecting them to heart attacks, they developed these intractable arrhythmias and even died, and you can see that on the quantification on the graph next to it. And interestingly, when you inhibited these kinases, the opposite happened, so that when you stressed them, they actually didn't have these arrhythmias and did quite well. And if you took heart cells from these, so those are action potential tracings, these are heart cells that were taken from these mice, even to the layperson, you'll appreciate that on the right side, which is the cardiomyocyte heart cell with activation of this kinase, the action potential is quite a bit broader. It's longer. So there's a prolongation of the action potential duration. And in fact, if you look at these cardiomyocytes long enough with patch clamping, you can see they get these spontaneous activity that's shown below. And these are what really lead to arrhythmias in patients who have these prolonged action potential durations. And so we next sought to understand why that was. And interestingly, it appears that there's a channel called the sodium channel that's important in depolarizing the cardiomyocytes. But if these channels don't close, you get a persistent sodium current, and that's called the late sodium current, quite important in conditions like long QT syndrome, particularly long QT3. And these cardiomyocytes all had about 4-fold increase in the late sodium current. And we went on to show that this was due to the fact that these channels get phosphorylated by this kinase that, again, is activated in conditions of stress. And we could block that late sodium current. You can see that in the two graphs below. And if you block the late sodium current, you can not only normalize the action potential duration, but you could get rid of these extra beats that you see that lead to these arrhythmias. So we had pretty good data that inhibiting this kinase blocked the late sodium current and decreased this arrhythmic potential when this kinase is activated. And so this sort of led to spam call. So this sort of led to a hypothesis that perhaps inhibiting this kinase might be important for those conditions where there's an increase in late sodium current. And this really led us to this long QT syndrome. We all know about long QT syndrome. We see patients with them. There are three major types. I'll really talk about long QT2 and 3 because those are the two types where there's actually an increase in the late sodium current. And this is just a graphic of what that looks like. And you can see that the QT interval, which is the surface ECG marker that corresponds to the action potential duration, is prolonged in these patients. And the corollary from this is that perhaps if we shorten that QT interval, there's pretty good epidemiological data that the longer your QT interval, the higher the risk of arrhythmias. If you shorten these QT intervals, that you would decrease this arrhythmic risk. So that's what the whole premise was based on. And in fact, if you take patients with long QT3 and you derive from their stem cells, you can derive iPSC, induced pluripotent stem cell-derived cardiomyocytes. They have prolonged action potential duration. And when you inhibit the kinase with a tool compound that we developed, you can shorten the action potential duration. And we went on to show that you can actually decrease these extra depolarizations, these triggers of arrhythmias. And so this was really the basis for formation of what used to be called long QT Therepeutics and then became Thryv Therapeutics. It was formed in 2019 with the usage IP from Beth Israel Deaconess, which is where I was at that time. And we were able to acquire a really potent series of SGK1 inhibitors from Sanofi and develop those into much more specific inhibitors with much less off-target effects. The company's done quite well with the Series A completed, and as I'll show you, we have some interesting results, both preclinically and clinically. So one of the first things we did, we talked to Mike Ackerman, who has a huge collection of these cardiac cells derived from patients with long QT1 and 2 and 3, and he tested these compounds that we developed to see that if we could actually shorten the action potential duration. And initially, we were really focused on long QT3, but from Mike's work and from work done with others in Europe, we found that not only in long QT3, but also in long QT2, you could actually shorten the action potential duration quite consistently with these inhibitors of SGK1. And in some cases, you could even do it in long QT1, although we've not quite focused on that. This is work done with a consortium that we work with in Europe, and these are various different models that are shown here. This on top is an iPSC cardiomyocyte model from patients with long QT2. You can see that again, we can shorten the action potential duration. We can get rid of these after depolarizations. This has now been done in 3 or 4 different families of long QT2, and it seems agnostic to the type of mutation that's there in long QT2, so that's good news. On the right-hand side is a rabbit model of long QT2. You can appreciate the really long action potential that's seen in these rabbits. When you treat these rabbits with the inhibitor, you can, again, pretty much normalize the action potential duration. And the last 2 graphs show a different approach. So obviously, this is an orphan disease, hard to recruit patients. And we thought, well, if you're seeing it in all kinds of long QT2, what if we pharmacologically block the channel that's responsible for long QT2, which is the hERG channel? So you can do that with a drug that's approved. We use it all the time to treat patients with atrial fibrillation called dofetilide, very specific blocker. You can do that, and you can prolong the action potential duration. And when you treat the cardiomyocytes or even the heart, so below is a guinea pig ex vivo perfused heart treated with dofetilide, prolongs the action potential, you treat it with an SGK inhibitor, and you can normalize the action potential. So a sort of pharmacological model of congenital long QT. And you can do this even in an in vivo model. So there's a canine model, anesthetized, treated with dofetilide. You increase the action potential duration. And when you treat these dogs in a short period of time with long QT inhibitor, you can, again, normalize the action potential duration. So this led to our first sort of proof of concept trial in patients. These were healthy volunteers who were brought in, treated with dofetilide, and then we treated them with the SGK1 inhibitor to see all the things we had seen in guinea pigs and dogs, that when we shortened their action potential duration, did we see that here as well? And in fact, so this is the study design. In the interest of time, I won't go over it in great detail, but each patient served as their own control. It's a thorough QT type study where patients were treated with dofetilide and the drug, washed out, and then just treated with dofetilide, so we could compare apples to apples, right? And this was done in 2 different ways. So either they had their placebo first and then the then the SGK1 inhibitor, or vice versa. So this is what those results showed. So if you took all patients, and one of the caveats of this study was that because we couldn't really give patients the drug to prolong it past 500 milliseconds, that's what the FDA approval is based on. We didn't have patients with really prolonged QT intervals. But what we saw is if you take all comers, we could shorten the action potential duration by about 10 or 15 milliseconds, depending on how you slice the data. And in fact, if you took the patients with the longer QT intervals, you could shorten it even more, which was quite along the lines what we were thinking, that you want a drug that is much more effective and your QT is much longer. If you take, and we'd previously done a study in normal people without dofetilide where we didn't overshorten. That was the other concern. So this was all good news. And if you, in fact, slice it by looking at each of those patients, you can see that those patients who had really prolonged QT intervals would shorten their QT by 40, 50 milliseconds, which was really quite meaningful. And those that had somewhat lower did so to a less degree. But the important point to note is that all those patients who were placed on the SGK1 inhibitor had QT intervals after treatment that were in a range that we wouldn't consider to be at a high arrhythmic risk. So having done that, we are now soon to actually look at results in patients, about 12 patients, proof of concept study with congenital long QT syndrome, long QT 2 and 3, that have been treated with 2 different doses of this inhibitors, a short duration study, about 4 days, with an endpoint looking at their QTC. And in all these patients, their QTC is more than 480, which is where we consider them to be at a high arrhythmic risk. And so, what we're going to hopefully unveil soon is whether the drug works in shortening their QT interval. So I'm, in the interest of time, I think I'm good, right? Good. So I'm going to stop there. We have a couple of other programs for atrial fibrillation and HFpEF, where interestingly, SGK1 also is activated, but I'm not going to talk about that, given this is an inherited arrhythmia symposium. And thank you very much.
Excellent. Well, next up, we have Dr. Howard Surks. He's the Chief Medical and Scientific Officer at Cardurion Pharmaceuticals. He's a physician scientist and cardiologist who ran a basic cardiovascular lab at Tufts Medical Center in Boston before spending the last 15 years in pharmaceutical drug development. So welcome, Dr. Surks.
Thank you very much. I really would like to thank the organizers of the meeting for inviting us here. It's both exciting and humbling to be here amongst people who are really on the forefront of treating patients with inherited arrhythmic disorders, and of course to be here with patients as well. So we really appreciate it. So I'd like to spend a few minutes telling you about our company and then focusing on one of our clinical programs, which is a small molecule inhibitor of calcium calmodulin-dependent protein kinase 2 for the treatment of catecholaminergic polymorphic VT. So Cardurion is a company based in Boston that has 2 clinical stage programs. We have an inhibitor of phosphodiesterase 9, which is currently in clinical trials for heart failure. And as I mentioned, we have a CAMK2 inhibitor, which we are exploring for multiple CV indications, including CPVT, and that's what I'm going to spend most of the time talking about. We are really focused on exclusively new drugs for cardiovascular diseases. The PD-9 inhibitor is currently in phase 2 proof of concept trials, and a CAMK2 inhibitor, as I'll describe later, is just initiated an initial trial in patients with CPVT. This is just a snapshot of our pipeline. As you can see, our most advanced program right now is our PD-9 inhibitor, and we have really focused a lot of our research activities on developing small molecule CAMK2 inhibitors. As I'll describe in a moment, this is a target, CAMK2, that's been pursued in the pharmaceutical industry for a long time, and we've, through just deploying a lot of force and medicinal chemists and biologists at this target for a number of years, have had some success on getting molecules that can be brought into clinical trials. And so we have both oral and intravenous inhibitors that we are developing for a range of cardiovascular disorders. So focusing in on the CAMK2 inhibitor program, my apologies to the electrophysiologists in the room for going through excitation-contraction coupling, but for everyone listening, and just to also put into context why CAMK2 is so interesting, I'll just spend a moment on this. We know, as everyone knows, depolarization of the cardiac cell membrane leads to opening of voltage-gated calcium channels and an influx of calcium into the myoplasm, and that calcium binds to and phosphorylates. That calcium binds to and opens the ryanodine receptor at step 2 on the slide, leading to a large amount of calcium flooding from the sarcoplasmic reticulum into the cytoplasm. That calcium binds to the troponin complex on the actin-myosin filaments, leading to actin-myosin cross-bridge cycling and contraction. In cardiac relaxation, that calcium has to be removed from the cytoplasm, either by actively pumping it back into the SR or pumping it out of the cell in exchange for sodium. Now, CAMK2 is a protein kinase. Its function is to phosphorylate its target proteins, changing their properties. And CAMK2, as the name would suggest, is a calcium-sensitive kinase. When the calcium levels rise during the contraction phase of the cardiac cycle, CAMK2 is activated. And when the calcium levels fall, CAMK2 becomes relatively inactivated. And CAMK2 can become hyperactivated in the setting of multiple diseases. And that generally occurs through post-translational modifications to the enzyme. And that can lead to disease because CAMK2 phosphorylates multiple proteins, many of them ion channels, in the cardiomyocyte. And that can lead, and when CAMK2 is excessively active or no longer calcium responsive, that can lead to inappropriate calcium conductance and arrhythmias. Importantly for CPVT, the catecholamine surge that occurs with exercise or emotional stress may activate CAMK2, and that may be a factor that could precipitate arrhythmias in patients with CPVT. That has been demonstrated in some animal models, which I'll mention in a moment, and is what we are currently investigating in CPVT patients. So CAMK2 has been a difficult target. As I mentioned, it's been sought after. CAMK2 inhibitors have been sought after for a number of years because there are so many non-clinical models that have demonstrated an important role in disease states. And we are fortunate to have the first one that has gone into human testing. And as I mentioned earlier, we are developing both oral and intravenous forms for different diseases. One of the reasons it's been so difficult to develop CAMK2 inhibitors is with most kinase inhibitors, the trick is to find a molecule that is sufficiently specific for that target and doesn't inhibit other targets. And of course, potency, so that a reasonable administered dose can have the appropriate effect on the molecule. And very importantly for CAMK2, it's important that the molecule not get into the central nervous system because CAMK2 plays a very important role in the brain in learning and memory. So one of the central tenets of our discovery process, our research process, has been to only focus on molecules where we can demonstrate that they are excluded from the brain. And this is just one example where in the figure on the left, you can see the plasma exposures in the clear bar and the minimal exposures in brain tissues to the right. And that's at a dose that is many-fold higher than what we administer to humans. So when in developing CAMK2 inhibitors for CPVT, we are really standing on the shoulders of giants. The role of CAMK2 in CPVT has been demonstrated by others, going back to Silvia Priori's study back in 2011, shown on the left, where she showed in a ryanodine receptor mutation knock-in mouse model that KN93, a CAMK2 inhibitor, could prevent the bidirectional ventricular tachycardia induced by epinephrine and caffeine. And more recently by Vasilios Bezarides and Bill Pugh. Dominic Abrams, by the way, was on that paper as well, in Circulation, which showed using a AAV transduced peptide inhibitor of CAMK2 that they could also prevent catecholamine-induced or programmed electrical stimulation-induced ventricular arrhythmias with CAMK2 inhibition. So we have now initiated studies with our specific and potent CAMK2 inhibitors. This figure just shows an example of the pharmacokinetics and how we can establish CAMK2 inhibitory activity in humans. On the left are the plasma concentrations, a lower and a higher concentration, and on the right are the associated levels of CAMK2 inhibition that we have detected using a novel CAMK2 activity assay from the blood, where you can see we can get down to almost complete inhibition of CAMK2 activity. And so we have now embarked on a clinical trial in patients with CPVT. We are doing that in collaboration with investigators who are in this room, who we are very grateful to for their advice, and also CPVT patients who we are also very grateful to, who have given us an idea of what it's like living with this disease, and also what would be feasible to do in terms of a clinical trial. And so the concept is an initial single-dose study. It's an intrapatient dose escalation. And these are in patients who have had a treadmill test that demonstrates that despite their standard of care therapy, whether it's beta blockers, flecainide, sympathetic denervation, that they still have some ventricular arrhythmic activity detectable. And then we are randomizing them to one of two sequences where each patient has 3 more exercise treadmill tests. Before each test, they either receive a placebo or one of 2 dose levels of the CAMK2 inhibitor. The overall schematic is shown here. Our initial trial is in 12 patients, initially in adult patients with CPVT. We are starting our investigation in patients who have known RYR2 mutations. It is quite possible that CAMK2 inhibition may demonstrate efficacy in other CPVT mutations, but at least in the initial study, we are beginning with a slightly more specific population, although RYR mutations are already a very diverse group. And we are conducting the trial at 11 sites in the US, Canada, Italy, and France. And then the schematic is, as you can see here, those are the patients who in screening who have demonstrable ventricular arrhythmic activity are randomized to one of 2 schedules where they come in 3 times for treadmill tests about a week apart, and each time they receive either dose A or dose B or dose C, which could be 2 dose levels of the inhibitor or placebo. And the main endpoint in the trial is the level of inhibition of exercise-induced ventricular arrhythmic activity. So just to summarize, we're excited to have the first CAMK2 inhibitor in human clinical development, and very eager to determine whether this molecule can potentially bring some help to patients with CPVT. We've been assessing its safety profile in phase 1 healthy volunteers. And we now have also demonstrated that we can inhibit CAMK2 robustly in humans. And our first trial in patients with CPVT is now underway. We have an optimized formulation of this molecule that we think that is clearly giving us good concentrations in the blood and is something that is easy to take and we can bring all the way through development and into patient use if it is successful. And of course, we are considering other indications where CAMK2 inhibition may be able to confer some benefit to patients. So again, I really want to thank everyone for inviting us here. We're really honored to be able to present to this group and participate in the symposium today. Thank you.
Thank you both for the excellent talks. In the spirit of today, we are over time, but we probably have time for we're okay? 10 minutes late. Okay. Perfect. Any questions for our speakers?
Don't be shy. There are all these questions this morning. We're over time. There are questions now.
I guess I'll ask a question. So in terms of the CPVT program, you mentioned that patients will need a pathogenic RYR2 variant. Now, we're always plagued by the dreaded VUS. So in patients who have a clear diagnosis, will they be eligible for your study?
Thanks. So initially, patients in this first 12-patient trial will require a clear known mutation that is associated with clinical CPVT. And that is really just because it's our very first study, and we want to begin with a very clear-cut population. But I do think that there is potential for patients with the clinical syndrome and you know, VUS mutations, as you noted, to benefit. And we hope that if we can demonstrate some activity in the initial population, that we will broaden it to include those patients you mentioned.
I just had a quick question. I was curious about the SGK1 inhibition, and it seemed to work in a bunch of different arenas, LQT1 and 2 and 3, and it all had the same effect. So why is it that is it just an effect on that sodium inward current? And is the secondary effect through sodium-calcium exchange where you decrease that current as well because there's less sodium inside the cell? Is that why it works in all three forms of?
Yeah, so it works. The most robust data we have is in long QT2 and 3. Long QT3 is pretty obvious because there is a late sodium current, and I think it changes the channel biophysics of these channels and prevents that. Long QT2 is actually interesting. We just, you know, the hypothesis has been when you prolong the action potential duration, as you point out, you have more sodium influx, and that secondarily leads to more calcium. In fact, there's actually pretty good data that even in long QT2, there's a large amount of late sodium current. For example, for these patients who have really prolonged QT interval in long QT2, you can treat them with mixilotine. More and more people are doing that, and you can shorten their action potential duration. You can decrease their arrhythmic risk. And now we have direct data that if you take, for example, the rabbit model or these iPSC cells with long QT2, they have a substantial increase in the late sodium current, and inhibiting SGK1 normalizes that. And so it doesn't act on these potassium channels, but what we think is happening is that actually CAMP kinase 2 gets activated in these. In fact, there's good data that when you prolong the action potential duration, either in long QT2 or even treating with dofetilide, you can actually activate CAMP kinase 2. And SGK1 actually happens to be a substrate of CAMP kinase 2. And so we've shown activation of SGK1. We think secondarily it might be through CAMP kinase 2. We haven't quite proved that yet. There are some experiments undergoing to prove it, but I think that's why SGK1 gets activated, and that's why it works. Why it doesn't work in long QT1 is not totally clear to me, and it works in some. There appears to be a mutation-specific phenotype there, but if someone has any ideas why it doesn't, I'd be curious to know.
Would sodium-calcium exchange inhibitors do the same thing as SGK1?
So the sodium-calcium exchangers would probably work upstream of that, right? So you could imagine that if you block this reverse mode, then you may not get activation of CAMP kinase 2, and maybe you get less late sodium current in these conditions other than long QT3. I think people in the past, I think someone from UCLA had really developed this to some degree. I'm forgetting his name now. But for some reason, they never took off in trials just because I think you'd have to inhibit that reverse mode of sodium-calcium exchange. And I'm not an expert on this, if somebody else might know. But those inhibitors were never, they worked fine in cell culture, but in vivo, there was always some issues. The genetic models were fairly convincing, I think, when they tried that, but I think for whatever reason that there weren't any good inhibitors to inhibit that specific mode of sodium-calcium exchange.
Thanks.
I have a quick question for both of you. So thanks so much, and I think it's great that there's targeted therapies being developed for CPVT and long QT. I remember when I was a medical student, and I learned about these really elegant physiology, and I was like, treat them with beta blockers. But with that said, beta blockers for long QT and beta blockers plus flecainide in CPVT work pretty well. In terms of what clinical trials do you guys envision doing in the future to show the world that we really need to be treating our patients with these new targeted therapies instead of the other medications that literally cost dollars a month? Have you guys thought forward about that?
Yeah, that's a great question. So as you said, beta blockers and flecainide are pretty cost effective for CPVT, and a lot of patients have good suppression of ventricular arrhythmic activity with those drugs. So we are targeting, at least for now, patients who don't have extremely good control on those medications. So it is a subgroup who we think still have unmet medical need. They're having events or they're having ectopy demonstrated on their treadmill tests at an unacceptable level, and there's a desire for more control of that. And our initial approach is to see whether CAM-K2 inhibition will provide more prevention on top of that, on top of those therapies, not in place of. But I think if that is demonstrated, the next approach would be to see whether a CAM-K2 inhibitor on its own would be better than those established therapies. And that would have to be very carefully done after demonstrating efficacy on top of those therapies in some sort of a withdrawal trial that we could envision in the future. But the initial approach is to not replace these drugs that are used and inexpensive, but to target the patients who, despite them, are still at risk.
Yeah, I think for long QT, there are a couple of arguments. One is the beta blockers work really well for long QT1, maybe modestly for long QT2, and not so well for long QT3. So long QT2 and 3 are the target we're first using. The second is, probably a lot of you in the audience have patients, young patients on beta blockers. And I can tell you that I have patients who even had syncope who refused to take beta blockers. So there's a compliance issue. I think there was a nice study from New Zealand, I think, that looked at this compliance and in patients with long QT, and it's somewhere in the 40% range, 40% non-compliance. So, and there is actually good data that when people come off the beta blocker, their arrhythmic risk is even higher. So I think if you could have a drug that doesn't have those side effects of beta blockers, and for all we know right now in the patients that we have dosed, they've really had no side effects on these drugs. And in fact, some of them have volunteered to come back for dose escalation studies. So I think that would be the argument. For long QT1, I think it's a trickier argument because beta blockers do work pretty well. But again, I think you could target some of those patients. For example, there are patients who are young kids, especially with sympathectomy, who are, so would this be a better approach than sympathectomy? I would argue that maybe so, if it's safe and it's durable. But that's sort of, for now, we all the patients who have been dosed have been on a beta blocker, apart from 2 who couldn't tolerate it. So the initial trial will be on patients with beta blockers.
Thank you, and everybody. I think with that, we'll close. Pam, do you have a burning question?
Well, patient perspective, perhaps, very quick. What kind of commitment does a patient, if I joined the CPVT trial, for example, what kind of commitment do I have to make as a patient? Is it over a long period of time? Is it a one-time deal?
Yeah, it's a great question. It is over a number of weeks. It's not an extremely long period of time, but it's not by any means an insignificant commitment, because it does require several exercise treadmill tests and to come back to the clinic each time to have them performed. So it is, you know, three treadmill test visits after an initial screening visit. And so, you know, we recognize that's a lot for patients, and we're always looking for ways to make it easier, make it more comfortable, it's a big focus on what we try to do, but it is a substantial commitment, we acknowledge.
Great.
Thank you, everybody.